another what you would consider as a side product, but then side products can then grab you and take you into new directions of research, and that is what is happening to us at the moment. We became very much interested in exploring the structure, the molecular background of some of the neuropsychiatric disorders that have been very difficult to approach before.
But I suppose knowing about the different kinds of neurons and the different kinds of receptors, you know, where are they placed on which neuron? I suppose that could lead you to new kinds of drug developments for these things.
Absolutely. I think today the major reason why there are no new drugs in the pipeline in most of the drug companies, and why especially those drug companies that focused on psychiatric disorders began to close down the research facilities, is because we don't understand the basic neuronal network mechanisms that underlie these disorders. And without further discovery research, there will be no new drugs available. And also what is important in neuropsychiatric disorders is prevention strategies. If you don't know what brings about these diseases, like what happens to the normal operations of the brain of an 18-year-old boy who was absolutely clever and happy, and then something happens in a very, very short time at a very strict period of development, and after that he ends up with schizophrenia. Unless we know what happens mechanistically and at the molecular level, there is no chance either for prevention or for a much more focused and targeted drug development.
So I suppose there has been a lot of focus for some years now on studies done with brain scans, because it's so fascinating that you can look into the living, thinking, feeling brain. But has that taken a little bit of attention away from that basic research into, so what are the cellular and subcellular mechanisms of the brain?
This is natural when there is a new technology that can be delivered at a reasonable cost to large number of laboratories, hospitals, then it takes over for a time. This is not unusual in either science or the development of society. And certainly it has soaked away funds from other directions, but it also parcellated the brain much more finely than we ever imagined. And it also enables scientists to look at the development of events in time, how each cortical area joins in at different time into a process that eventually leads to behavior. So it's extremely important, and with the picking of all the easy fruit that was available, the realization then came that within all those areas, joining in at different time, there are defined sets of nerve cells joining it at different time themselves, and that's where the next chapter and big push is. And of course, we have been aware of this need for decades, but now it's internationally recognized, and perhaps this is also why we received this prize.
Do you think that this prize given for this type of work that you three have been doing can stimulate a kind of interest among students in this kind of work?
There are other people who like dynamic. Those people who come out from physics, engineering background, they know there is a problem and they like working on problems. And we have a series of problems. One of them is how causation occurs. What I like to refer to is reciprocal causation, because there is no easy way to go from one area to the other, or from one cause to the other. Just going back to what Peter said, for me, every new window into the brain is a welcome progress. So the fMRI is extraordinary new type of new way of looking at brain activity. But again, indeed, if you look at the time resolution, they say, what is perhaps new here, and it's what we can offer, is an alternative to drug research, for example. Schizophrenia and other mental diseases have been classified as diseases of a particular receptor in the past. That's a rigid approach. For those people who are coming from mathematics and engineering, they said, it has to be a dynamic. Something has to happen in time. And indeed, what happens in time is the kind of rhythms that we can provide, because these rhythms hold together systems, separate systems. I can identify a system called the limbic system by a particular rhythm. So if I develop a drug, or I test an existing drug, and I know that affects this particular rhythm, I know which part of the brain is being affected by this. In fact, we tested an interesting so-called thalamocortical rhythm, and every single psychiatric drug that exists, that it's in the market, had a tremendous effect on this particular rhythm, but left relatively intact all the others. So this is a very robust phenotype that cuts through evolution. As I mentioned, the mouse and the human brain is exactly the same. So this is a good target to see how different drugs affect dynamic.
You're all working on the hippocampus. Why the hippocampus? You could choose to work on any cortical area, I suppose.
Indeed, and Tamás and I started to work on the visual cortex and worked for almost a decade together on the visual cortex, both here in Hungary and in Oxford, and made significant discoveries. But the more we discovered, the more we realized how many different players are in the visual cortex and how many distinct streams of visual information flow through, and each cell only participates in one or the other, and this is then integrated in other visual cortical areas, of which there is a very large number. So I eventually myself realized that my life is just not going to be long enough to define all the cell types in the visual cortex, and drifted into the hippocampus, first testing the presence of exoaxonic cell that was still jointly with Tamás, and once I found it, I realized this is just as good area to figure out how different inhibitory cells modulate the activity of the principal pyramidal cells, which are organized into a single sheet in the rodent.
For me, it was primarily for two reasons. One, the person responsible is Gyuri, who introduced me to the wonderful world of oscillations in the hippocampus. And for me, it was very fascinating to ask the question how the structure is organized to explain this theta rhythms in the hippocampus. The other one was ischemia. I was always interested in the pathological aspects of our research. And since the hippocampus is the most vulnerable area in the brain in ischemia and also in some parts of epilepsy, that was another reason why I thought it would be very beneficial and interesting to exploit our knowledge we gathered in the visual cortex to the hippocampal circuits. And then I think we made quite a number of interesting discoveries jointly with Gyuri in the field of ischemia, correlating again the structural abnormalities to function.
I inherited the hippocampus from my mentor, and I inherited the first rhythm from my mentor, who discovered, he didn't discover theta, but he discovered this 5 per second rhythm in behaving animals. So he was the first person who ever recorded from behaving animals during some interesting learning task and said, oh, there is this prominent oscillation. That was a good moment for me because of his dynamic, but it was also a good moment in retrospect because since then I worked with the thalamus, neocortex, cerebellum, basal forebrain, multiple areas, but I always return to the hippocampus, and I do it because I realize that it is a structure so far away from the inputs. There are multiple steps away from your eyes and from your ears, and whatever you do with the outside world, the hippocampus just doesn't listen. It just goes on its own. And so this is probably what allowed me, and perhaps collectively us, to begin to understand the brain from inside out. And I think it is a more objective way that anything we see is relevant, whereas if we are going from outside in with lines, vertical and horizontal and edges, and hope that you will put together vision one day from the simplicity, probably won't happen, or won't happen in this way, or will take a long time. But understanding the brain from within, taking the brain as the independent variable and see how the brain classifies the world is an eye-opening moment. It was an eye-opening moment for me. I spent about the first 8 years of my life being an ethologist. I was very much was impressed by the work of Konrad Lorenz and Tim Berry, and there were unending debates whether a particular behavior was this type or that type, and it couldn't be solved experimentally without going into the brain. The moment we went to the brain, we could say, yes, this is a consumatory behavior, this is an appetitive behavior, this is a gnostic behavior, this is another type, because the brain determines, the brain classifies how it perceives. The brain is a reader of its own activity, and the brain translates the external world according to its own will.
And this is where oscillations come in.
This is where oscillations comes in, if I can take a moment. So what happens in evolution is that the way how we think is that vision is about inputs coming into your brain, and the brain is ready and just like this camera will register the information. No. The way it works is that first the brain figures out how to move the eyes. So it generates an output how to optimize the conditions, how a camera, such as the eye, can see. And then after the optimization, it uses a short period of time that allows that with the smallest possible energy, it will capture the information it is looking for. Same thing with touch, same thing with taste. You move your tongue when you would like to taste something. There are transient periods right after this output generated by the brain that the brain is sensitized. Now, this was inherited, this principle was inherited by the rest of the brain. So instead of the hippocampus listening all the time to the neocortex, we figured out it's the other way around. The hippocampus is the reader, the hippocampus is the receiver, hippocampus is the listener to the neocortex, but the hippocampus starts the negotiation. That is, with its oscillation, it coordinates the activity of multiple areas of the cortex to say, okay, in the next 100 milliseconds, I will dedicate my entire attention to whatever you send to me. So in the brain, the communication is not like in the Shannonian information theory that there is an ever-ready receiver, but the receiver is the one that starts the communication, and this is where brain rhythms are indispensable.
Then how has your work on oscillations and understanding oscillations and how they come about impacted on what we know about memory formation?
I'm glad you brought this up. One of our most important contribution probably to neuroscience is to show how information is put into small packages and send it from the hippocampus to the neocortex while we are asleep. We have known it for a long time that sleep does some service to our cognitive function. It was always suspected that it is probably REM sleep, the famous dream sleep, that is important. What we found is that if you would like to produce plasticity very effectively, you have to bring together powerfully and synchronize them, as Tamás emphasized. And I found a pattern in the hippocampus that happens when we are not attentive And this is a synchronous pattern. It's a package that lasts about 100 milliseconds associated with